Antisense oligonucleotides, ASOs, represent a pivotal advancement in molecular medicine, offering a powerful tool for modulating gene expression with precision and specificity. Through targeted intervention in genetic pathways, ASOs provide a therapeutic strategy for a myriad of diseases that were previously considered intractable at the genetic level. The design of ASOs involves a sophisticated array of strategies, each tailored to maximize efficacy, specificity, stability, and safety, and minimize off-target effects and toxicity. Optimal design and chemical modifications. The journey of designing an effective ASO begins with the careful selection of the target mRNA sequence, a process that leverages advanced computational tools to predict accessibility and binding sites. Chemical modifications play a critical role in this narrative, enhancing the ASO's pharmacological profile by improving stability against enzymatic degradation, increasing binding affinity, and reducing immune responses. From backbone modifications such as phosphorothioates and locked nucleic acids, to sugar modifications like 2-pan-O-methyl and nucleobase alterations, each chemical adjustment is a strategic decision meant to optimize the ASO for its intended purpose. Chimeric and conjugate innovations. The innovation does not stop at single-type modifications. Chimeric designs, including gapmers and mixmers, integrate multiple modification types within a single ASO to exploit the synergistic effects, achieving a delicate balance between activating RNase H and maintaining high binding affinity. Furthermore, conjugation strategies, such as attaching to molecules like GalNAc or incorporating lipid and peptide elements, have opened new avenues for targeted delivery, enhancing the uptake and specificity of ASOs to particular tissues or cells. Future directions and challenges. Despite the impressive strides in ASO technology, challenges remain. The synthesis of complex chimeric and conjugated ASOs presents both technical and economic hurdles. Moreover, ensuring the safety, efficacy, and delivery of ASOs across different biological barriers and within diverse patient populations continues to be an area of active research. Regulatory pathways for these novel therapeutics also demand careful navigation as the clinical implications of long-term and systemic ASO use are further understood. A promising horizon. The development of ASOs is a testament to the remarkable progress in genetic and molecular therapies. As research continues to push the boundaries of what is possible, the role of ASOs in treating not just rare genetic disorders but also common diseases broadens, heralding a new era of precision medicine. With ongoing advancements in ASO technology and an increasing understanding of human genetics, the potential for ASOs to transform medicine is not just promising, it is imminent. Understanding antisense oligonucleotides, ASOs. Before discussing modifications and design strategies, it's important to understand what ASOs are. Think of ASOs as small customized snippets of DNA or RNA that are designed to bind to messenger RNA, mRNA. This binding can prevent the mRNA from producing proteins, essentially silencing a gene. Imagine ASOs as precise molecular scissors or blockades that can specifically target and modulate gene expression. Types of synthetic and biochemical modifications. The modifications to ASOs are crucial for enhancing their stability, binding affinity, and overall effectiveness. These can be broadly categorized into backbone modifications, nucleobase modifications, and conjugations. Backbone modifications. Antisense oligonucleotides, ASOs, undergo various backbone modifications to enhance their therapeutic potential by improving stability, cellular uptake, and specificity while reducing off-target effects and toxicity. Below is a comprehensive list of these modifications, along with a description and an assessment of their advantages and limitations in vivo. Backbone modifications for ASOs, phosphorothioate, PS. Description. In this modification, a non-bridging oxygen in the phosphate backbone is replaced with a sulfur atom. This slight change significantly increases resistance to nucleases, which are enzymes that could degrade the ASO. Advantages. Enhanced binding to plasma proteins increases ASO stability in circulation and improves cellular uptake across various tissues. Limitations. PS modifications can cause non-specific interactions with proteins, leading to potential side effects. They can also activate the immune system unintentionally. Peptide nucleic acids, PNAs. Description: PNAs replace the entire sugar phosphate backbone with a peptide-like backbone. This radically alters the structural nature of the nucleic acid. Advantages: extremely high stability and strong binding affinity to complementary RNA or DNA sequences. Limitations: due to their unnatural backbone, PNAs have poor solubility and require effective delivery systems. They are also not recognized by cellular enzymes, complicating their pharmacokinetic and pharmacodynamic profiles, morpholino oligomers. Description: morpholino oligomers have a backbone made of morpholine rings linked through phosphorodiamidate linkages. They do not contain a traditional sugar or phosphate group. Advantages: excellent resistance to enzymatic degradation and no activation of the immune response due to their neutral backbone. Limitations: challenges in delivery inside cells due to their neutral and bulky backbone. They also have high synthesis costs. Locked nucleic acids, LNAs. Description: LNAs incorporate a methylene bridge connecting the 2Z oxygen with the 4-carbon of the ribose ring, locking the ribose in the 3D endoconformation. Advantages: high thermal stability and increased affinity for target RNA, allowing for shorter ASO sequences that are still highly effective. Limitations: potential for hepatotoxicity with high doses and risk of off-target effects due to strong and specific binding. Phosphanoacetate and phosphorodithioate. Description: these modifications involve changing the linking groups in the backbone to alter the charge and steric hindrance. Phosphorodithioate replaces both non-bridging oxygens with sulfur. Advantages: improved nucleus resistance and altered interaction with proteins, potentially reducing non-specific binding. Limitations: can still induce some immune responses and may have solubility issues depending on the extent of modification. Thiophosphate. Description: similar to phosphorothioate, this modification replaces one of the non-bridging oxygens with sulfur, but is placed at different positions to tune properties. Advantages: provides a balance between nucleus resistance and binding affinity without overly increasing non-specific interactions. Limitations: the overall physicochemical properties can be unpredictably altered, affecting solubility and distribution. Additional backbone modifications for ASOs. 2-O-alkyl modifications. Description: modifications like 2-O-methyl, 2-OMe, and 2-O-methoxyethyl, 2-MOE, involve modifying the 2-as position of the ribose sugar in the nucleotide. While technically a sugar modification, it significantly affects the backbone's overall properties by influencing flexibility and enzyme resistance. Advantages: these modifications improve nuclease resistance and increase affinity for the target RNA. They are also less immunogenic compared to other modifications. Limitations: they can still have off-target effects and may require careful optimization to balance stability and efficacy. Bridged nucleic acids, BNAs. Description: BNAs, which include locked nucleic acid, LNA, derivatives, have a bridge that locks the ribose in an ideal conformation. This group also includes other types such as 2-DAS, SOUT4-BNA, and tricyclo-DNA, which modify the sugar ring differently. Advantages: they offer excellent stability and affinity, with high specificity for targeting RNA. They also allow for the design of shorter ASOs due to their high binding affinity. Limitations: the strong binding affinity can lead to toxicity if not properly controlled, and the cost of synthesis can be higher. Ethylene bridge, ENAs. Description: ethylene nucleic acids, ENAs, contain an ethylene bridge between the 2-Sals oxygen and the 4-carbon of the ribose. This is similar to LNAs but uses a different kind of chemical bridge. Advantages: they provide strong nucleus resistance and increased binding affinity to RNA. Limitations: like LNAs, the high binding affinity may increase the risk of off-target effects and toxicity. Phosphoramidate, PMO. Description: phosphoramidate linkages replace the phosphate group with a linkage that contains nitrogen, altering the charge and interaction profile of the backbone. Advantages: these modifications lead to neutral backbones that do not support RNase H activity, but are excellent for steric blocking applications, e.g., in splice-switching oligonucleotides. Limitations: they often require specific delivery mechanisms due to their neutral charge and may have limited tissue distribution. Phosphotriester. Description: phosphotriester modifications involve replacing one of the non-bridging oxygens with a group that can be cleaved enzymatically, potentially used for prodrug approaches. Advantages: provides protection from nucleases until conversion into an active form in the target site. Limitations: the stability and delivery of these compounds can be challenging, and the cleavage efficiency may vary depending on the enzyme availability in target tissues. Choosing the right modification. The selection of a backbone modification depends on several factors, including the desired mechanism of action, e.g., RNase H activation, steric blocking, the target tissue, the need for duration of effect, and the safety profile. The development of ASOs is often a balance between optimizing therapeutic efficacy and minimizing adverse effects, necessitating a nuanced approach to the design and choice of backbone modifications. Other innovative modifications. Beyond the common backbone modifications I've outlined, there are several other innovative modifications that have been explored to improve the properties of antisense oligonucleotides, ASOs. These modifications aim to optimize therapeutic efficacy, reduce toxicity, and enhance delivery and stability. Innovative backbone modifications for ASOs. Amide-linked oligonucleotides, AMNA. Description: this modification involves replacing the phosphodiester linkages with amide linkages, transforming the backbone into a more peptide-like structure. Advantages: amide linkages can significantly enhance the stability of oligonucleotides against enzymatic degradation and improve biocompatibility. Limitations: the synthesis can be more complex, and the biophysical properties such as flexibility and hybridization dynamics can be altered, which may affect the targeting efficiency. Sulfone backbone. Description: introducing sulfone groups into the backbone provides a polar but uncharged linkage, differing from the negatively charged phosphates. Advantages: sulfone-modified ASOs are more resistant to enzymatic degradation and can exhibit better pharmacokinetic properties due to their neutral charge. Limitations: the impact on the duplex stability with RNA needs careful evaluation, as it might affect the ASO's efficacy. Boranophosphate. Description: this modification replaces a non-bridging oxygen in the phosphate group with a BH3 group, creating a boranophosphate linkage. Advantages: it enhances nuclease resistance and can improve the overall stability of ASOs. Limitations: there are concerns about potential toxicity and the in vivo stability of the borano groups, as well as their interaction with biological systems. Fluoroarabinonucleic acids, FNA. Description: modification of the sugar moiety to contain a fluorine atom at the 2 position in an arabino configuration. Advantages: increases binding affinity to RNA and provides higher nucleus resistance. Limitations: the synthesis of FNA-modified oligonucleotides is complex, and the impact on RNA binding needs to be carefully balanced to avoid off-target effects. Selecting a modification strategy. The choice of a specific backbone modification is driven by the goals of a particular therapeutic application. Factors such as target tissue, desired duration of action, route of administration, and safety profile play critical roles in the decision-making process. Researchers often employ a combination of different modifications in a single ASO to harness the benefits of each while mitigating their limitations. This combinatorial approach can help tailor the pharmacokinetic and pharmacodynamic properties of ASOs to meet specific clinical needs. The field of ASO development is continuously evolving, with ongoing research aimed at discovering and optimizing new backbone chemistries to improve therapeutic outcomes. Additional innovative backbone modifications for ASOs. Cyclic phosphate. Backbone. Description: this modification introduces a cyclic structure within the phosphate backbone, aiming to restrict flexibility and enhance stability. Advantages: improved resistance to nucleases due to the cyclic structure, which makes it more difficult for enzymes to cleave the backbone. Limitations: the rigidity imposed by the cyclic structure can impact the ASO's ability to hybridize with its target RNA, potentially reducing efficacy. Siloxane backbone. Description: replaces the phosphodiester linkage with a siloxane linkage, silicon-oxygen bonds. Advantages: this modification provides exceptional thermal stability and resistance to enzymatic degradation. Limitations: the siloxane backbone may have different interaction properties with cellular proteins and RNA targets, which can affect delivery and efficacy. Alkyl chain backbone. Description: introducing alkyl chains, such as methyl or ethyl groups, into the backbone to modify interaction characteristics. Advantages: enhanced lipophilicity, which can improve cellular uptake and membrane permeability. Limitations: overly hydrophobic modifications might lead to aggregation in biological fluids and nonspecific binding to cellular components. 3-ose nucleic acid, TNA. Description: TNA involves replacing the natural ribose or deoxyribose sugar with a 3-ose sugar, creating a novel backbone structure. Advantages: TNAs are highly resistant to nucleus degradation and can bind strongly to both RNA and DNA targets. Limitations: the synthesis of TNAs is complex and costly, and their biological compatibility and delivery mechanisms are not as well understood. Glycol nucleic acids, GNA. Description: this is a modification where the sugar backbone is replaced with a glycol unit, creating a simpler and more stable linkage. Advantages: GNAs show high thermal stability and resistance to enzymatic degradation, with potential applications in environments where chemical stability is crucial. Limitations: GNAs may have reduced flexibility and might not be as effective at natural RNA-based pairing, requiring further structural optimization. Strategic considerations: the selection of backbone modifications is influenced by the specific requirements of the ASO's intended application, including target specificity, resistance to degradation, immune response, and delivery method. Each modification carries its own set of advantages and challenges, making the design of ASOs a highly tailored and strategic process. As research progresses, we continue to see the development of new backbone chemistries that aim to address the limitations of existing modifications while enhancing the therapeutic potential of ASOs. The continuous exploration and understanding of these innovative backbones are crucial for advancing ASO technologies and expanding their application in medicine. Exotic backbone modifications for ASOs. Xenonucleic acids, XNAs. Description: XNAs are synthetic nucleic acid analogues, where both the sugar and the phosphate backbone are replaced with alternative molecular structures. Types of XNAs include HNA, hexatyl nucleic acids, CINA, cyclohexanyl nucleic acids, and FNA, furanose nucleic acids. Advantages: these novel structures can exhibit enhanced stability, nucleus resistance, and unique binding properties. XNAs also explore the potential for increased diversity in molecular recognition and catalytic capabilities. Limitations: XNAs are challenging to synthesize and require specialized enzymatic tools for their manipulation. Their interaction with biological systems is not fully understood, potentially limiting their immediate therapeutic application. Zwitterionic oligonucleotides. Description: this modification involves introducing zwitterionic groups, which contain both positive and negative charges within the same molecule, into the backbone of the ASO. Advantages: zwitterionic modifications can improve solubility in water, reduce nonspecific binding, and potentially enhance biocompatibility. Limitations: the balance of charges might affect the overall conformation and hybridization properties of the ASO, impacting its efficacy and specificity. Nucleic acid nanostructures. Description: this approach uses the principles of DNA origami to fold ASOs into specific three-dimensional structures designed to enhance cellular uptake and targeted delivery. Advantages. Nanostructuring can be used to create highly specific delivery vehicles for ASOs, increasing localization to target tissues and reducing off-target effects. Limitations. The complexity of design and production, along with the need for precise control over folding and stability in biological environments, poses significant challenges. Carbon backbone oligonucleotides. Description. In this experimental modification, the entire phosphate sugar backbone is replaced with a carbon-based structure, creating a backbone that is entirely hydrocarbon-based or containing other elements like silicon. Advantages. This could theoretically provide extreme resistance to enzymatic degradation and unique physicochemical properties not seen with natural nucleic acids. Limitations. Synthesis can be extremely complex and costly. The biological compatibility and functional efficacy of such dramatically altered molecules are largely untested. Light-activated oligonucleotides. Description. Incorporating photolabile groups that can be cleaved or activated by light allows temporal and spatial control over ASO activity. Advantages. This enables precise control over the therapeutic activity of ASOs, allowing them to be activated only at specific sites and times within the body, minimizing systemic exposure. Limitations. Requires the development of safe and effective methods to deliver light to target tissues, which can be challenging, especially for internal organs. These highly experimental modifications are at the frontier of nucleic acid research and represent a mix of conceptual and technical innovations. Each modification aims to overcome specific limitations of current ASO technologies, such as stability, specificity, and delivery challenges. As these technologies mature, they could potentially open new avenues for genetic and cellular therapies. Assessment of in vivo performance. The challenge with these modifications lies in balancing efficacy with safety. For instance, while PNAs and morpholinos offer high stability and strong binding, their unnatural backbones complicate their in vivo application due to issues with cellular uptake and potential toxicity. Phosphorothioate oligos are more drug-like with good tissue distribution but can engage with non-target proteins, leading to side effects. Each backbone modification also impacts how an ASO is metabolized and cleared from the body, with some modifications like PS leading to longer circulation times, but also potential accumulation in non-target tissues like the kidneys. The choice of backbone modification is thus crucial and must be tailored to the specific therapeutic need, target tissue, and required duration of action, balancing these factors against the risk of side effects and immune responses. Nucleobase modifications. Nucleobase modifications in antisense oligonucleotides, ASOs, are crucial for enhancing their binding affinity, stability, and specificity, while reducing off-target effects and toxicity. These modifications are made directly to the bases, adenine, cytosine, guanine, thymine, or uracil in RNA, of the nucleic acids. Here's a comprehensive overview of known nucleobase modifications, their descriptions, and an assessment of their advantages and limitations in vivo. Common nucleobase modifications for ASOs. 5-methylcytosine, 5-MC. Description: methylation of the cytosine base at the 5th carbon position. It's a naturally occurring modification in DNA. Advantages: enhances stability and binding affinity to the target RNA. Can also reduce immune stimulation. Limitations: the effect on binding specificity can vary, and overmethylation might interfere with the desired regulation of gene expression. Pseudoridine. Description: a modified version of uridine where the connection between the ribose and the uracil base is changed from a carbon-nitrogen bond to a carbon-carbon bond. Advantages: increases the thermal stability of ASO RNA duplexes and improves translation efficiency in mRNA therapies. Limitations: can be more costly to produce. The increased stability might enhance the risk of off-target effects. Inosine. Description: a modification where adenine in the DNA or RNA is deaminated to produce inosine, which pairs with cytosine, adenine, and uracil. Advantages: broadens the scope of base pairing, potentially enhancing target binding under certain conditions. Limitations: can sometimes cause wobble-base pairing, leading to reduced specificity and potential off-target effects. N6-methyladenosine, M6A. Description: methylation of adenine at the nitrogen 6 position, a modification commonly found in RNA. Advantages: can affect the structure and function of RNA, potentially influencing ASO binding and efficacy. Limitations: the biological impact of M6A in ASOs is less studied, and the specific effects can depend on the cellular context. 2-thyridine. Description: incorporation of a sulfur atom at the second position of the uridine base. Advantages: improves the thermal stability of ASO RNA duplexes and increases resistance to enzymatic degradation. Limitations: the modification might affect the natural processing of RNA and can alter the dynamics of RNA interference mechanisms. Bridged, ultra-bridged bases. Description: bases that are modified to include bridge structures, e.g., ethylene or methylene bridges, across the base to rigidify or alter stacking interactions. Advantages: can significantly enhance base stacking, stability, and specificity of ASO binding to the target. Limitations: the complexity of synthesis and potential impacts on natural base pairing properties could limit their use. Additional nucleobase modifications for ASOs. 5-propenyluridine and 5-propenylcytidine. Description: these modifications involve the substitution of the hydrogen at the 5 position of uridine or cytidine with a propenyl group. This change enhances base stacking interactions and increases the melting temperature of the ASO RNA duplex. Advantages: they can significantly increase binding affinity to target RNA and enhance nuclease resistance. Limitations: the strong binding affinity might sometimes lead to off-target effects, and the modification can be more costly to synthesize. 2-deoxy-2-Y-fluoro BD-arabinonucleic acids 2-FNA. Description: in this modification, the 2-hydroxyl group of the ribose sugar is replaced with a fluorine atom in an arabinose sugar configuration. Advantages: provides increased binding affinity and exceptional nucleus resistance while maintaining good specificity. Limitations: the synthesis process can be complex, and the fluorine atom might interact unpredictably with cellular components. Qcene. Description: Qocene is a naturally occurring modified nucleoside found in tRNA, where the base guanine is modified. Advantages: this modification can be used to explore novel interactions with RNA and potentially enhance translational fidelity or modify RNA function in targeted ways. Limitations: limited research on its incorporation into ASOs and its effects on RNA behavior in vivo, which means its practical applications are still not well understood. 7-deoxyadenosine and 7-deoxyaguanosine. Description: these modifications involve replacing the nitrogen atom at the 7 position of the purine ring with a carbon, which prevents the formation of toxic adducts and improves the chemical stability of purine nucleosides. Advantages: enhances the stability of nucleic acids against degradation and reduces the formation of secondary structures that can interfere with binding. Limitations: altered hydrogen bonding properties can affect the overall hybridization characteristics of the ASO. Pyrazolopyrimidine analogs. Description: these are synthetic bases that mimic the structure of natural bases, but are designed to enhance specific base pairing capabilities. Advantages: can improve the selectivity and affinity of ASOs for their target RNA, potentially allowing for more effective gene silencing. Limitations: their synthesis and incorporation into ASOs are complex, and their long-term stability and toxicity profiles need thorough investigation. These additional nucleobase modifications are part of ongoing efforts to optimize ASO design for therapeutic applications. Each modification offers unique advantages that can be tailored to specific therapeutic needs, but they also bring challenges, particularly in synthesis, stability, and potential biological interactions. Cutting-edge nucleobase modifications. The field of nucleic acid chemistry continues to explore a wide range of additional nucleobase modifications. These modifications are aimed at enhancing the properties of antisense oligonucleotides, ASOs, for improved therapeutic applications. Here are a few more cutting-edge nucleobase modifications that are currently being investigated. Hypoxanthine. Description. Hypoxanthine is a naturally occurring nucleobase derived from the deamination of adenine. In ASOs, its incorporation can mimic the behavior of both guanine and adenine. Advantages: provides versatile base pairing options, potentially increasing ASO's affinity and specificity for various RNA targets. Limitations: the ambiguity in base pairing could also lead to mismatches and off-target effects if not precisely controlled. Universal bases, e.g., nitroindole difluorotoluene. Description: universal bases are designed to bind with any of the 4 standard bases, A, T, G, C, with reasonable affinity but without strong discriminatory binding, used to reduce sequence-specific binding constraints. Advantages: they can simplify the design of ASOs by allowing for more flexible base pairing and can help in reducing the impact of sequence variations or mutations on ASO binding. Limitations: the non-specificity of binding can potentially increase the risk of off-target effects and reduce overall efficiency of the ASO. Isocytosine and isoguanine. Description: these are modified versions of cytosine and guanine with slightly altered hydrogen bonding patterns, designed to enhance pairing fidelity and chemical stability. Advantages: they can provide improved thermal stability to ASO RNA or ASO DNA duplexes and enhanced resistance to enzymatic degradation. Limitations: their chemical synthesis and incorporation into oligonucleotides can be complex, and the biological implications of these modifications are still being fully understood. Base modifications for enhanced fluorescence. Description: modifications like attaching fluorescent groups to nucleobases can be used to track and monitor the distribution and localization of ASOs in cellular or in vivo systems. Advantages: useful for research and diagnostic purposes, allowing visualization of ASO dynamics and interactions within biological systems. Limitations: the addition of bulky fluorescent groups can affect the biophysical properties of the ASO, potentially altering its binding characteristics and cellular uptake. C5 modified pyrimidines, e.g., 5-bromouracil. Description: chemical modification at the C5 position of pyrimidine bases, used to enhance specific properties like nucleus resistance or to introduce cross-linking capabilities. Advantages: these modifications can be used to increase the stability of ASOs or to create more robust interactions with target RNA. Limitations: depending on the modification, it can increase the synthesis complexity and cost and might affect the ASO's natural interaction with cellular enzymes and proteins. The development of new nucleobase modifications is an active area of research that continually evolves as new discoveries are made in chemistry and molecular biology. These innovations are driven by the need to overcome specific challenges associated with ASO therapies, such as improving target specificity, reducing toxicity, and enhancing delivery mechanisms. These modifications are just a snapshot of the possibilities within the realm of ASO development. Assessment of in vivo performance. The choice of nucleobase modification in ASOs is typically tailored to achieve a balance between enhanced biological stability and therapeutic efficacy while minimizing off-target effects and toxicity. In vivo, these modifications often provide crucial improvements in pharmacokinetics and pharmacodynamics, such as increased resistance to nucleases and improved binding to target RNA sequences. Advantages: overall, nucleobase modifications generally enhance the stability and functionality of ASOs, making them more effective in therapeutic applications. They can also help in evading immune detection and reducing the activation of immune responses. Limitations: however, the modifications can also bring challenges such as potential changes in the natural processing of RNA, increased risk of off-target effects due to enhanced stability and binding affinity, and the need for more complex and expensive synthesis processes. The development of ASOs with nucleobase modifications is a dynamic area of research, with ongoing studies aimed at optimizing these molecules for clinical use. Each modification needs to be evaluated not only for its chemical and physical properties, but also for its biological implications, including interactions within the complex cellular environment. Conjugations, GalNAc, conjugates, tethering ASOs to N-acetyl-galactosamine helps target them specifically to the liver, enhancing the delivery of ASOs to hepatocytes. Picture a delivery truck specifically designed to only deliver goods to one particular warehouse. Conjugation modifications in antisense oligonucleotides, ASOs, are critical for improving their delivery, stability, and efficacy in therapeutic applications. Conjugation involves attaching specific groups or molecules to ASOs to enhance their pharmacological properties. Below is a detailed list of known conjugation modifications, along with descriptions of each and an assessment of their advantages and limitations in vivo. Known conjugation modifications for ASOs. Cholesterol conjugation. Description: cholesterol is covalently attached to the ASO to improve its association with cellular membranes and facilitate uptake by cells through passive absorption. Advantages: enhances cellular uptake, particularly in liver cells, and can improve biodistribution and pharmacokinetics. Limitations: may lead to non-specific binding and accumulation in non-target tissues, potentially causing off-target effects. GalNAc, N-acetyl-galactosamine conjugation. Description: GalNAc is attached to ASOs to specifically target and enhance uptake by the Asia-log-glycoprotein receptor, ASGPR, on hepatocytes, making it highly liver-specific. Advantages: greatly enhances liver specificity and uptake, improving therapeutic index for liver-targeted therapies. Limitations: limited to liver targeting, does not aid in targeting other tissues or cells lacking ASGPR. Pegylation, polyethylene glycol conjugation. Description: attaching polyethylene glycol PEG molecules to ASOs to increase their solubility, stability in circulation, and to reduce renal clearance and immunogenicity. Advantages: extends circulation time and can reduce immune recognition and clearance. Limitations: pegylation can sometimes hinder tissue uptake and cellular entry, and high molecular weight PEG can trigger anti-PEG antibodies, leading to reduced efficacy over time. Peptide conjugations. Description: conjugation of peptides to ASOs to facilitate cell-specific targeting and uptake, particularly using cell-penetrating peptides, CPPs, that enhance delivery across cell membranes. Advantages: can target specific cell types and enhance cellular uptake significantly. Limitations: stability of peptide ASO conjugates can be an issue, and there may be variability in efficiency depending on the peptide, ASO sequence, and target cell type. Antibody conjugations, antibody-drug conjugates, ADCs. Description: ASOs are conjugated to antibodies that target specific cell surface receptors, enabling receptor-mediated endocytosis. Advantages: provides very specific targeting capabilities to cell types or tissues expressing the target antigen, potentially enhancing therapeutic efficacy and reducing off-target effects. Limitations: complex to design and manufacture, can be costly, and the stability of the conjugate in vivo needs to be optimized. Folate conjugation. Description: attachment of folate to target cells expressing the folate receptor, commonly used to target cancer cells that overexpress this receptor. Advantages: facilitates targeted delivery to cancer cells, enhancing therapeutic specificity and reducing impact on normal cells. Limitations: limited to targeting cells that express the folate receptor. Variability in receptor expression can affect delivery efficiency. Lipid conjugations. Description: conjugation of various lipid molecules to ASOs to improve membrane interaction and uptake, similar to lipid nanoparticles. Advantages: enhances the overall delivery of ASOs into cells, especially useful for tissues with high lipid affinity. Limitations: can potentially increase the risk of lipid-related toxicities and may affect the distribution pattern of the ASO. Assessment of in vivo performance. The choice of conjugation depends on the specific therapeutic goals, target tissues, desired duration of action, and safety profile. These conjugation strategies can significantly impact the in vivo behavior of ASOs. Advantages: enhanced delivery, increased stability in biological fluids, targeted cell or tissue uptake, and reduced immunogenicity. Limitations: complexity in synthesis, potential for unexpected biological interactions, off-target effects, and sometimes increased cost and manufacturing challenges. The ongoing development in the field of ASO conjugation is aimed at overcoming these limitations while maximizing therapeutic potential. Additional conjugation modifications for ASOs. There are additional conjugation modifications for antisense oligonucleotides, ASOs, that expand their functional capabilities and enhance their pharmacological profiles. These modifications can help address specific challenges such as targeted delivery, tissue penetration, and clearance rates. Here are some further conjugation strategies that are used or being explored in the development of ASOs. Vitamin conjugations. Description: conjugation of vitamins such as vitamin B12 or vitamin E to ASOs. These vitamins can serve as ligands for receptors on target cells, facilitating targeted delivery. Advantages: enhances specific uptake by cells expressing the vitamin receptors, potentially increasing the therapeutic index and reducing systemic side effects. Limitations: the effectiveness can be limited by the expression levels of the vitamin receptors on target tissues, and there may be competition with natural ligands. Aptamer conjugations. Description: conjugation of aptamers, which are oligonucleotide or peptide molecules that bind to a specific target molecule. Aptamers can be designed to target specific proteins or cell surface markers. Advantages: provides a high degree of specificity for targeting, with the potential for modulating the activity of target molecules directly. Limitations: requires careful design to ensure specificity and stability of the conjugate, and the complexity of synthesis can be high. Polymer conjugations. Description: attaching polymers other than PEG, such as polylactic acid, PLA, or polyglutamic acid, which can help in forming a protective envelope around the ASO. Advantages: these polymers can improve the stability and solubility of ASOs and adjust the release rates of the ASOs in biological environments. Limitations: the biodegradability and immunogenicity of the polymers need to be carefully evaluated to prevent adverse reactions. Small molecule conjugations. Description: conjugation of small molecules that can either enhance the targeting of ASOs or modulate the biological environment to improve ASO efficacy. Advantages: this can include enhancing crossing of the blood-brain barrier or modulating the immune response to increase tolerance and efficacy of ASOs. Limitations: potential for increased toxicity, off-target effects, and challenges in precisely controlling the pharmacodynamics of the conjugate. Metal chelation. Description: conjugation of metal chelating groups that can bind to metal ions, which can be used for imaging or therapeutic modulation. Advantages: allows for the tracking and imaging of ASO distribution in vivo, and potentially the use of metal ions for therapeutic effects. Limitations: the presence of metal ions can lead to toxicity if not carefully controlled, and the chelation may affect the stability and function of the ASO. These additional conjugation modifications provide researchers with a wide array of tools to enhance the delivery and function of ASOs in vivo. Each conjugation strategy has its own set of advantages that make ASOs more effective for specific applications, but they also introduce challenges that need to be addressed to ensure safety and efficacy. Developments in conjugation technology continue to evolve, driven by advances in chemistry, biology, and medical science. Cutting-edge and experimental conjugation modifications for ASOs. The research into antisense oligonucleotides, ASOs, is continually advancing, with cutting-edge and highly experimental conjugation strategies emerging to tackle challenges in delivery, specificity, and efficacy. Here are some innovative and experimental conjugation modifications being explored. Clickable chemistry for in situ conjugation. Description: utilizing bioorthogonal click chemistry that allows for conjugation reactions to occur within the biological environment without interfering with normal cellular processes. This approach can be used to attach various functional groups to ASOs after administration. Advantages: provides the ability to control where and when the conjugation occurs, potentially improving the targeting and reducing off-target effects. Limitations: the complexity of the reaction and the need for precise control over the reaction conditions in a biological environment pose significant challenges. Nanoparticle conjugates. Description: conjugating ASOs to nanoparticles such as gold nanoparticles, quantum dots, or polymeric nanoparticles to facilitate cellular uptake and provide protection against degradation. Advantages: enhances cellular uptake, allows for controlled release, and can provide imaging capabilities to track distribution and localization. Limitations: nanoparticles can sometimes elicit immune responses, and there are concerns about the long-term biocompatibility and toxicity of these materials. Photoresponsive conjugates. Description: incorporating photoresponsive groups that can be activated or deactivated by specific wavelengths of light, allowing temporal and spatial control over ASO activity. Advantages: enables precise control over the therapeutic activity of ASOs, reducing systemic exposure and potential side effects. Limitations: requires the development of safe and effective methods to deliver light to target tissues, which can be particularly challenging for internal organs. Targeted delivery systems using exosomes. Description: conjugating ASOs to exosomes or engineering exosomes to carry ASOs, utilizing the natural cell-derived vesicles for targeted delivery. Advantages: exosomes can naturally target specific cells and are generally well tolerated, reducing immunogenicity and potentially enhancing delivery across biological barriers like the blood-brain barrier. Limitations: scalability of exosome production and characterization, as well as ensuring the stability and integrity of the ASOs during and after exosome encapsulation are ongoing challenges. Dual ligand targeting. Description: using two different ligands in a single conjugation strategy to target two distinct receptors on the target cells, potentially enhancing specificity and uptake. Advantages: increases targeting specificity and can reduce off-target effects by ensuring that ASOs are delivered to cells expressing both receptors. Limitations: complexity in design and synthesis, potential for increased cost, and the need to balance the binding affinities of both ligands. MRI contrast agents. Description: conjugating ASOs with MRI contrast agents to enable real-time imaging of ASO distribution and dynamics within the body. Advantages: provides valuable information on the pharmacokinetics and localization of ASOs, assisting in therapeutic monitoring and optimization. Limitations: the addition of contrast agents can alter the pharmacodynamics of ASOs and may introduce imaging-related challenges or toxicity. These experimental approaches represent the forefront of research in ASO technology, each with the potential to significantly impact how genetic diseases are treated. However, these methods also bring new challenges in terms of safety, efficacy, and delivery, requiring thorough investigation and optimization before they can be widely adopted in clinical settings. Design strategies. Design strategies for ASOs are primarily focused on maximizing their therapeutic potential while minimizing off-target effects and toxicity. These include length optimization. Typically, ASOs are 18 to 21 nucleotides long. This length provides a balance between specificity for the target, mRNA, and efficiency in terms of cellular uptake and gene silencing. Strategies for optimal length design. Empirical testing and iteration. The theoretical predictions for optimal ASO length must be validated empirically. This involves synthesizing ASOs of varying lengths, targeting the same mRNA region, and testing their efficacy and specificity in cellular or animal models. Iterative testing helps refine the length based on observed performance, balancing between efficacy, specificity, and safety. Use of bioinformatics tools. Bioinformatics tools can predict mRNA secondary structure and accessibility, guiding the choice of target sites and influencing the optimal length. These tools help identify regions of the mRNA that are likely exposed and available for ASO interaction, suggesting where longer or shorter ASOs might be more effective. Hybridization energy calculations. Calculating the hybridization energy for ASO mRNA pairings of different lengths can help predict which lengths will form the most stable and effective interactions. Higher hybridization energy generally correlates with more robust binding, informing decisions on length optimization. Safety and efficacy profiling. Beyond theoretical and empirical optimization, ongoing safety and efficacy profiling is essential. This involves monitoring for any adverse effects in preclinical and clinical trials, which might be influenced by ASO length and the extent of modifications. Target site selection. Choosing the right segment of mRNA to target is critical. This involves bioinformatics analyses to predict mRNA secondary structures and accessibility, ensuring the ASOs bind effectively. Target site selection is a foundational aspect of designing effective antisense oligonucleotides, ASOs. This step is critical because it determines how specifically and effectively the ASO can interact with its intended mRNA target, influencing both the efficacy and safety of the therapeutic intervention. Here's a detailed overview of the strategies and considerations involved in selecting target sites for ASOs. Key considerations in ASO: target site selection, accessibility of the target site, RNA structure. mRNA molecules often form complex secondary structures such as loops, bulges, and stems. ASOs need accessible regions within these structures to bind effectively. Computational tools and experimental methods like SHAPE, selective 2-hydroxyl acylation analyzed by primer extension, can be used to map accessible sites on the mRNA. Protein binding. Regions of mRNA that are bound by proteins may be less accessible to ASOs. It's crucial to avoid targeting these regions unless displacement of the protein is desired as part of the mechanism. Specificity of the target sequence. Sequence uniqueness. The target sequence must be unique to the mRNA of interest to avoid off-target effects. Bioinformatics databases and tools like BLAST are used to ensure that the selected sequence does not have significant homology with other transcripts in the genome. Conserved regions. Targeting conserved regions within a family of related genes might be advantageous for conditions where multiple isoforms or closely related proteins need to be downregulated. Efficacy of target engagement, GC content. The GC content of the target site affects the stability of the ASO-mRNA duplex. A moderate GC content is ideal, as high GC content can make the duplex too stable, complicating dissociation, while low GC content may lead to unstable interactions. Location within the mRNA. Targeting regions near the start codon or within the coding sequence can be more effective than targeting the untranslated regions, UTRs, depending on the ASO mechanism, e.g. RNase H-mediated degradation versus steric blocking. Strategies for optimal target site selection. Computational prediction. Use bioinformatics tools to predict mRNA secondary structures and identify potential accessible sites. Tools can also assess sequence conservation and potential cross-reactivity with other mRNAs. Energy modeling can help predict the stability and dynamics of ASO-mRNA interactions, aiding in the selection of sites that balance stability and accessibility. Empirical validation, in vitro screening, synthesize and test multiple ASOs targeting different regions of the mRNA to identify the most effective sites empirically. Techniques such as luciferase reporter assays can be useful for this purpose. In vivo testing. Once promising target sites are identified in vitro, they should be validated in vivo in relevant animal models to assess the pharmacokinetics, efficacy, and safety of the ASO. Use of high-throughput technologies, massive parallel screening. Technologies such as high-throughput sequencing and microarray-based assays enable the simultaneous screening of thousands of potential target sites across various conditions and cell types. CRISPR-Cas9 screens. Adapting CRISPR technology to screen for effective ASO target sites by systematically knocking down potential target regions and assessing cellular outcomes. Integration with delivery considerations. The selected target site should also be compatible with the chosen delivery strategy. For instance, if the ASO is intended for liver-specific delivery, the target mRNA should be highly expressed in the liver, and liver-specific considerations should influence site selection. Target site selection is a multi-step process that blends computational prediction, empirical validation, and strategic consideration of biological contexts. Each step is critical to ensure that the chosen ASO will achieve its intended therapeutic effect while minimizing the risk of off-target effects and other unintended consequences. The evolving understanding of RNA biology and advancements in technology continuously refine these strategies, enhancing the potential of ASOs in genetic medicine. Chemical modification patterns, strategic placement of modifications like PS or 2E MOE, can dictate the binding strength and resistance to degradation. The pattern of alternating modifications might be used to fine-tune these properties. Chemical modifications are crucial in designing effective antisense oligonucleotides, ASOs. These modifications not only improve the ASO's pharmacokinetic and pharmacodynamic properties, but also enhance its stability, binding affinity, and specificity, while reducing off-target effects and potential toxicity. Here's an in-depth look at the strategies and considerations involved in selecting chemical modification patterns for ASOs. Strategies for ASO chemical modification patterns. Balancing modification types. The choice and pattern of modifications are designed to balance the ASO's ability to hybridize with its target mRNA, its stability against enzymatic degradation, and its overall toxicity profile. A common strategy is to use a combination of backbone and sugar modifications to achieve these objectives. Gapmer design, a popular approach where central nucleotides of the ASO are left unmodified or lightly modified to retain the ability to activate RNase H, while the flanking regions are heavily modified for increased stability and affinity. This design maximizes therapeutic activity while minimizing exposure of the RNase H active center, reducing off-target effects. Uniformly modified ASOs. In scenarios where steric blocking, rather than RNase H-mediated cleavage, is desired, ASOs may be uniformly modified across their entire length. This can be particularly useful in applications like splicing modulation. N-group modifications. Modifications at the termini of ASOs can provide additional protection against exonucleases and can be tailored to improve cellular uptake or tissue-specific targeting. For example, adding cholesterol or other lipid molecules can enhance uptake by hepatocytes in the liver. Optimizing for target and tissue. The modification pattern can also be optimized based on the expression profile of the target mRNA and the specific tissues involved. For example, liver-targeted ASOs often use GalNAc conjugation to enhance uptake via the asialoglycoprotein receptor. Considerations for modification patterns. Biophysical properties. Modifications affect the ASO's hybridization properties, such as melting temperature, Tm, and the formation of secondary structures. These must be carefully calibrated to ensure effective hybridization without compromising biocompatibility and cellular uptake. Pharmacokinetics and distribution. The modifications influence how ASOs are absorbed, distributed, metabolized, and excreted in the body. Optimal modifications can enhance these properties to ensure that the ASO reaches and persists within the target tissue at therapeutic concentrations. Immunogenicity and toxicity. Some modifications might reduce the risk of activating immune responses or causing off-target effects, which is crucial for clinical applications. The design of chemical modification patterns in ASOs is a sophisticated process that requires a deep understanding of molecular interactions, the biological environment, and the therapeutic goals. Advances in chemical biology and an expanding array of modification options continue to refine this process, allowing for the creation of more effective and safer ASOs tailored to specific diseases and conditions. Use of chimeric designs. Combining different types of modifications in a single ASO, e.g., a gapmer, where a central region of DNA is flanked by modified nucleotides, offers a balance of stability and efficiency. Chimeric designs in antisense oligonucleotides, ASOs, represent an advanced strategy to optimize the therapeutic potential of ASOs by combining different chemical modifications within a single molecule. This approach allows for the exploitation of the unique properties of each type of modification to achieve a synergistic effect, enhancing efficacy, stability, and specificity while minimizing toxicity and off-target effects. Here's an in-depth look at the strategies, purposes, and considerations involved in the use of chimeric designs for ASOs. Purpose of chimeric designs in ASOs. Chimeric ASOs are designed to enhance binding affinity and specificity. By combining modifications, chimeric ASOs can achieve higher affinity and more selective binding to the target mRNA. Improve stability. Different modifications can protect the ASO from enzymatic degradation from both endonucleases and exonucleases. Modulate pharmacokinetics and dynamics. Tailored modifications can influence how ASOs are absorbed, distributed, metabolized, and excreted in the body. Reduce immunogenicity and toxicity. Strategic modifications can decrease the likelihood of ASOs being recognized by the immune system as foreign, thus reducing potential side effects. Key strategies in chimeric ASO design. Gapmer design. Structure. Gapmers are chimeric ASOs that contain a central segment of DNA, the gap flanked by segments of chemically modified nucleotides, the wings. Purpose: the DNA gap segment enables the recruitment of RNase H for mRNA cleavage, while the modified wings enhance nucleus resistance and improve binding affinity. Common modifications. Wings are often composed of 2-O-methoxyethyl, 2-MOE, locked nucleic acids, LNAs, or phosphorothioates, PS, depending on the required properties. Mixmer design. Structure. Mixmers combine different types of modifications along the length of the ASO without the distinct gap and wing structure seen in gapmers. Purpose. This design allows for a finely tuned balance between nucleus resistance, binding affinity, and the ability to activate RNase H. Application. Useful in situations where a more uniform distribution of properties along the ASO is desired, such as when targeting regions with complex secondary structures. Conjugate chimeras. Structure. These chimeric ASOs are conjugated with other molecules like peptides, lipids, or carbohydrates to improve cellular uptake or target specificity. Purpose. Enhancements in delivery efficiency to specific tissues or cells, e.g., hepatocytes with GalNAc conjugation, or improve pharmacological properties. Example: ASOs conjugated with cell-penetrating peptides, CPPs, to facilitate crossing of cellular membranes. Considerations for chimeric design. Optimal placement of modifications. The effectiveness of a chimeric ASO depends on the strategic placement of each modification type. Computational modeling and empirical testing are crucial to determine the best arrangement for achieving desired therapeutic outcomes. Chemical synthesis challenges. Synthesizing chimeric ASOs with multiple modifications can be technically challenging and costly. Advanced synthetic techniques and thorough characterization are necessary to ensure batch consistency and efficacy. Biological and functional testing. In vitro and in vivo testing are essential to validate the predicted benefits of chimeric designs. These tests assess not only the therapeutic efficacy but also the potential toxicity and immunogenicity of the ASO. Regulatory considerations. The introduction of complex chimeric designs can pose regulatory challenges, as each component and its impact on safety and efficacy must be thoroughly documented. The use of chimeric designs in ASOs represents a sophisticated approach to overcoming some of the limitations of simpler ASOs. By combining various chemical modifications, researchers can create highly specialized and effective therapeutic agents tailored to specific diseases and conditions. This strategy is at the forefront of personalized medicine, leveraging molecular design to achieve precise therapeutic interventions with minimal adverse effects. Conclusion. Antisense oligonucleotides, ASOs, represent a groundbreaking approach in the field of genetic medicine, providing targeted strategies to modulate gene expression at the molecular level. By specifically silencing or modifying the translation of genetic information, ASOs offer immense potential for treating a vast array of diseases, particularly those with genetic foundations. The success of ASOs hinges on meticulous design strategies that encompass target site selection, chemical modifications, and the innovative use of chimeric and conjugate designs. Strategic insights and design considerations. The efficacy of ASOs is rooted in their ability to accurately target and bind to specific mRNA sequences, which requires precise identification of accessible and specific target sites. This is complemented by sophisticated chemical modifications that enhance the stability, efficacy, and safety of these molecules. Modifications such as phosphorothioates for backbone stability, 2-TED-O-methyl for increased affinity, and advanced chimeric constructs enable tailored pharmacokinetic and pharmacodynamic profiles suitable for various therapeutic needs. Advancements and future outlook. The continued development of conjugation strategies, like GalNAc for liver-specific targeting, and innovative delivery systems, further amplify the potential of ASOs to become mainstream treatments. Despite challenges in delivery, stability, and off-target effects, ongoing research and clinical trials are paving the way for ASOs to address not only rare genetic disorders but also common diseases, offering new hope where traditional treatments have fallen short. In conclusion, antisense oligonucleotides are at the cutting edge of personalized medicine, with the power to transform the treatment